Okay, so first and foremost, let me try and get the link for you guys. We have an evaluation of every single workshop. So I just want to request from students that during the workshop or after, if you could just take 5 minutes. I don't necessarily speak for 2 hours. I don't believe in speaking for 2 hours because no one has the attention span. So I'll speak for about 45 minutes to an hour. So if you could take the time to fill out the workshop evaluation, that would be really grateful, because a system can only improve if we understand what postgraduate students need. I know there's a lot of students that need help with quantitative data analysis, especially when you're at the stages of writing up your thesis and submitting. So we do have that. In second semester, you can set up a consult with me to guide you through the whole quantitative data analysis using either GraphPad or SPSS. So yeah, let's get started. So first and foremost, I'm going to put the, here we go, the evaluation in, so you can load that as we go along, and just take 2 minutes to fill it out. Be really grateful for that. And yeah, let's get started. Because we're only about 8 students, not like the usual 40 or 50, you can just keep it informal. I'll just ask you to have your mic, obviously, and your video, housekeeping rules. Yeah, my details do appear at the end, so I will send that. Everything appears, don't worry. Yeah, keep your video off, and I will keep mine off the entire time so that for bandwidth purposes, because it is 1 o'clock and it tends to slow down. The internet tends to slow down at campus. Okay, so let's do GLP and calculations. There are a lot of examples. Well, yeah, kind of a lot. So if there's something you don't understand, I think because we're only 6 or 7 students, you can just unmute yourself and then we can go back. But I've kept them very basic, because what I want students to understand is that if you understand conversions, then you will find calculations to be much easier. And so we're going to do a quick example of how, if a postdoc or a supervisor tells you to go into the lab and prepare a buffer, where do I start? Because I think a lot of students get lost in translation, and often we tend to panic unnecessarily. So yeah, so I'm going to just share my screen. I just hope my laptop keeps up today. Okay, so that's the evaluation that you guys can fill out. And here we go. Just take this out. Okay. Yeah, you see, this is, here we go. Okay, so welcome. For those of you who don't know who I am, my name is Aasiyah. I'm the academic facilitator at EPGS. I don't know if this is your first time attending our, sorry about that, attending our workshops. So we do various workshops. For those of you, it's the first time. We do various workshops on literature review, gathering your data, types of research, quantitative data analysis and collection. That's coming up for honors. We do it for masters and PhDs. We handle NRF and so on. So I'm the academic facilitator for natural sciences, and I focus on first-year PhDs, master students, and honors in the natural science and community health science faculty. Okay, so today we're doing the GLP and calculations. So before we get to calculations, we'll be focusing on understanding the process that is described as scientific method. Types of research, I'm just going to touch base on it quickly. Brief look at implementing good laboratory practices in basic scientific research, because I think a lot of students seem to struggle with this, which then inundates and annoys, so to speak, the senior students. And then we're going to look at data analysis. I'm just going to quickly discuss that and what is going to be offered. So we're not focusing on that. So for those of you who have registered via the student portal, you can register for data collection and data analysis, which is the May workshops. And the reason why we've pushed them late in the year is because honors students attend a lot of modules and they're only starting their research now. So it doesn't make sense for you to have a workshop now on how to do data analysis when you actually don't have data and you're trying to learn a lot of laboratory techniques. And then a brief thing on understanding your research group and setting, and the fact that any scientific practice, if you belong to a research group, it's essentially a collaborative process. And then we're going to touch base on basic calculations. And with the calculations, I'm not going to go in depth because that is your supervisor and your research group's job. I'm going to look at the basics that help you if you're struggling, because remember, we're a support system. So that's looking at dilution factors briefly, your basic calculations that are very, very important for buffers and compounds. And then the one thing that students really struggle with is standard curves and why they're important. And if you're working in certain fields and you're working with compounds or nanoparticles or something and so on, you need to always get comfortable with standard curves. And this is stuff you're going to use throughout. So that's why we'll be focusing on that as well. Okay, and any questions, feel free to either unmute or the chat box. I'll try and check it, and then we'll have a Q&A at the end. So as I said, this is the third workshop. The first two were more on literature review and academic writing and so on by my colleague. And now I'm focusing on calculations, data collection, and data analysis. So on the 9th of May, it's at 1 o'clock, and the 25th. So if you follow those, I think by the 9th, you should have a bit of data. So then we can look at how is data collected properly, outliers, how do you clean up? I think a lot of students don't understand is that when you're in lab and you collect a whole lot of data, you're using a plate reader, you're calculating and so on and so forth, you have to first clean up your data. But if you understand how you're going to collect your data, you know how to clean up your data and then eventually analyze your data. And then that becomes easier. And then on the 25th, we'll then slowly get into data analysis. So that is obviously your graph pad and your SPSS, which software to use, regression analysis, dosage curves, so on and so forth. So feel free to register for those. I have a lot of students that have registered late. You go to your student portal and you register via your student portal. So if you enter your student portal, your student number and your ID number, you go to workshops and courses, and then you can register for these that are coming up. Okay, so scientific method, this is where it all starts. I think the problem, and I've always said this to my honors students, is that honors is a steep learning curve. You know, there's a lot to do in a very, very short space of time, and it becomes extremely overwhelming. And the problem with that is that you are expected to write, you know, and we're coming from the mentality of, especially now with online, you're coming from the mentality of, I want to cram as much of information as possible to get the CAM so that I can write the exam and get to the next level. Now, when you come into postgraduate studies, it's slightly different. You're going to slowly start thinking independently. And the lucky thing about being in honors is that it's a steep learning curve, yes, but you are assigned a senior student. You are assigned a postdoc or someone who has been doing this before. So they guide you through that, but it's still a lot because you are required to attend modules. You're required to learn things on your own, which you've been doing. You're required to sometimes carry out experiments after you've been taught for a period of time, to carry out experiments on your own. In addition to that, you need to then make sure you understand what you're doing for yourself, as well as learn to work independently. So hence I said it's an iterative process, because when you come to scientific research, it's a step-by-step. So the wing it days are kind of over because you need to have a step-by-step. So that's where you ask a question and then you apply it. And then you try again and again, because you will find for those of you who've entered lab already, you're not going to get the same data set. So this is the key thing to remember, why we do a step-by-step series of workshops to guide you instead of jumping ahead and bombarding you with information. It's that you start with literature review. I need to understand what I'm doing. If you belong to a particular research group, it's either oncology, it's neuro, it's nano, it's chemistry, and each of those is divided into its own sector. Chemistry has organic chemistry, inorganic chemistry, nanochemistry, nanophysics, so on and so forth. You need to understand the literature that applies to your research group and your PI and so on. And then in addition to that, how am I actually going to navigate the question that I'm trying to ask, collect the data, analyze the data, and then one thing that's required of you, and I mean, it's not as difficult as, let's say, master's, PhD, and so on, it's understanding interpretation of that data in the context of literature. So unfortunately, you cannot get away with cramming information, but then this is where it becomes like a double-edged sword. It becomes problematic because we all overread, and then you get anxious, everyone wants to sound smart, and so on and so forth. So the key thing to understand is that you're going to make an observation. Remember, at honors level, you're given a slightly small project, you're assigned as a person. The idea is to then ask a question. In some cases, and remember, we're looking at community health sciences as well, they have an hypothesis. It's very, very important. And as a result, based on the hypothesis within social work or nursing, you're making a prediction, especially if you have epidemiological studies, you're going to make a prediction based on the hypothesis. Now, some of us in wet lab don't necessarily do that, but it's good to understand the process. And then you carry out the experiment a couple of times, and then you test it by analyzing the data. And then from that, you're able to then have a correct hypothesis or an incorrect. If you're doing wet lab, you won't necessarily have a hypothesis. You would have a particular compound. You're testing the particular compound in a particular, with a particular angle. And then from there, you're trying to see whether the compound has a, I don't know, upregulation of a gene, downregulation of a gene, anti-cancer, pro-apoptotic, so on and so forth, immunomodulating, antioxidant. There is so much that you can be looking at. And then you're setting out. But the idea is to ask a question, carry out the data collection, the experimental work, and analyze, report, and try again. And this mentality is something that is going to continue from now right until PhD and then some. And in industry as well, it's the same concept. Okay, so what do? I have a suspicion of exposure, and I think that maybe my exposure might influence an occurrence or an outcome. So I make it based on clinical practice. In some cases, when you're in wet lab, it's not based on clinical, because not all of us are in a clinical practice. We're in wet lab. It's based on a postulation. It's based on previous studies. And this is the thing I always explain to students. You need to understand there's absolutely no need to reinvent the wheel. I can guarantee you that someone has done what you're trying to do before. They have done it. The beauty of being an honest student is that you don't have to have a novel project. You can, but you don't have to. It's not required of you. Even at master's level, it's not required, because essentially what you're doing is you're introducing yourself to research at honors. At master's, you're mastering skills. It's at PhD and at postdoc, it gets more tricky because the requirement for publication is there. However, even though we have that, I always advise students to start thinking outside the box from the get-go. It makes it so much easier. And this doesn't apply just for the natural sciences. It's in the community sciences, which fall under us as well. If you're able to code from a younger age, okay, in the beginning, at high school level, at first year, at second year, you're already comfortable with coding. I can guarantee you, as you move up the ranks, coding will become completely so easy for you. And that's the same thing with laboratory work. So the idea is you're having an observation based on that, and you have a disease and an outcome. In our case with wet lab, you're observing this in your research that's been done. Either research that's been done previously in your lab, either via the guidance of a senior student, and then from there, you're going to make theoretical speculations. And this all falls under critical thinking. And a lot of students struggle with that. And I understand being online, it's very difficult. The idea is to learn to think outside the box. That is how research becomes more and more comfortable for you, is to learn to speculate. In order for you to do that, you have to critically think. If I read an article, I look at what they're doing in my lab. Do I understand what they're doing? What is the outcome? Why am I doing what I'm doing? If I'm making a nanoparticle, why am I making this nanoparticle? What is the end game? What data am I going to collect? And that comes with comfortability of reading. Okay, so that's your basic question. We're all answering a basic question. Exposure, outcome, easy. Disease outcome, or an outcome. I'm exposing cell lines to a certain compound, and I want to see the outcome of those, and then look at the association between them. That is the basic question we're constantly asking in research. That's where you start. So I think I just want to get students familiar with that before we get into how does it all fit together. Because if you think outside the box, trust me, the entire process of reading, writing, understanding, presenting, calculations, data analysis, all comes together. You unfortunately have to put it all together, and it's a lot. It is a lot to do on your own. So now that we understand, okay, I'm in lab, I understand what I'm doing, I understand why I belong to a particular research group, I know what's expected of me, I've been given articles by a postdoc or a supervisor to read up, so I know what I'm doing. So I belong to the neuro group, and I want to see whether the gold nanoparticle is going to affect my neuroblastoma cells, because I want to see maybe the pathway for Alzheimer's. I'm in a cancer lab, similar concept, breast cancer. I want to see if the upregulation of the BRCA1 gene or the downregulation will really influence the outcome of you getting, or the likelihood of you getting breast cancer. You see? So that's where you start. Where do I start? I need to first start with lab work. I have my cell lines. I need to extract. I need to either clone the gene, whatever you're doing. We start thinking outside the box, which means I need to get comfortable with good laboratory practice. And these things seem obvious, and that's the thing. Common sense is not as common as we think. And I think in academia, that's where we get it wrong, especially as senior students and supervisors, is that we think that a lot of these things are common sense. They're not, because no one told you. And that's the frustrating thing when you're in honors, is that everyone expects you to know this, but no one actually takes the time to teach you. No one says, this is how I collect good notes. If you do this, you can streamline it. And that actually helps you not get overwhelmed. So when you're not overwhelmed, you're able to focus better. Okay, so good laboratory practice. It sounds easy. I think a lot of this is just, Let's just all be decent and respect people's, but let's just go through it quickly. So good laboratory practice, it's actually a 400 and something document, but we're not interested in that. I just want students to understand good laboratory practice mustn't be confused with standards of laboratory safety. Okay? Good laboratory practice means from the time I enter the lab, when I am being taught a particular technique, I'm running through lab work. How am I actually planning it, performing it, monitoring it, and reporting it and archiving it? That's what good laboratory practice is. It's not just for the lab, but it's also for yourself. And this is the thing, we all struggle with, and we all have our ways in which we do it. So it applies to non-clinical studies. We all have our way of doing it, but don't confuse that with wearing a lab coat. That's not what we're talking about. That is something I think at honors level, at third year, we all know that already, that when you enter the lab, closed shoes, lab coat, so on and so forth. That's not what we're talking about. Good laboratory is if you learn from the get-go when you step into a lab to plan your week, to collect your data, to make up your buffers on a Monday or a Tuesday, to perform the experiments, to monitor the experiment, to check if you've made a mistake, and then write it all down properly, report it and archive it. I can guarantee you when you're writing up, it will streamline everything for you. So that's what we're trying to get students to understand. So what is it? It was created in the 70s. Like I said, there's a whole lot of documents. Why we're not interested in the history, we're just interested in how we're going to apply it. I mean, we can do the history, but I mean, come on, how many of us are actually going to hear me when I say that? It's just history, we don't care. I'm trying to streamline it as quick as possible. So it was invented in the 1970s because when they looked at in-depth investigations of toxicology labs, they discovered that a lot of people have poor laboratory practices, fraudulent activities, and you see this. As you join a lab, I think most of you have finished your advanced techniques course, so you've now been assigned various research groups. You enter the lab, you'll see this with your senior students. You think they know more, but sometimes they don't. As you learn, you realize, actually, my colleagues are a lot. They actually don't. They don't clean up after themselves. They don't sterilize the bench. They don't enter the lab and make a good workspace for themselves. Stuff like that, I can guarantee you, those small things contribute. Especially for some of you, I mean, like myself, that I'm in a molecular lab where you're doing PCR, where you're doing extraction of DNA and RNA. Something as simple as not wiping your bench down with DNA's away or DNA's thing can affect your sample. So you see, it's a compound effect. So that's the thing, poor laboratory practices where you have that. Some cases, equipment is not calibrated. And again, if equipment is not calibrated, you could easily, if you're doing a quantifying of your DNA, if you don't do proper serial dilutions, and this is the thing I'm trying to get students to really understand, if you don't do proper serial dilutions, you will get poor DNA yield. And then you'll have to go back in the lab and spend 6 weeks, 6 months, and most students have done this, 6 months to a year, only to find out that the equipment wasn't calibrated, or you didn't dilute your sample properly, or you didn't set the equipment properly, and you were getting a reading of 10 micrograms when you needed a reading of 1 microgram. You see what I mean? Something like that. Incorrect accounts of lab study. That is something that we think is so obvious to students, but it actually isn't. When we say incorrect or inaccurate accounts of actual lab study, it's in your laboratory book, write down everything. I can guarantee you're not going to remember. It doesn't matter how brilliant you were. It doesn't matter when you graduated cum laude. It doesn't matter with even your PhD. It's the fact that you're going to forget this, I can guarantee you. So every day when you're in lab, you enter with a good laboratory practice of planning, performing, monitoring, reporting, and archiving. So that is where incorrect accounts of your actual lab study will be avoided. So why? And the reason for this, like I mentioned, they looked at about in the 1970s, 40 toxicology labs, and they actually found that there was a need for good laboratory practice manual. And this is where I want students to understand is that it's not just about droning on, about writing your book, read a lot, learn to focus. It's not that. It's the fact that you have to understand the bigger picture. Something as small as pipetting the incorrect volume will waste 3 weeks of your time. If you can just pipette properly, calculate properly, understand the difference between a 1-in-2 dilution versus a 1-in-4 dilution, it will avoid you wasting time and getting frustrated. So one of the reasons was they found with the industrial lab known as BioTest. So it was a lab that was actually responsible for toxicology studies. And what happened with this lab is that it was under investigation because it ran a whole lot of testing in your pesticides, in your skincare, and so on and so forth. And it was running these big tests for all these drug companies. It was even Syntax, it was testing a drug. And what they found out, that what they were doing is that the data that they were generating on these particular studies was actually fraudulent. So what they were doing, it was a compound effect, because what was happening is that they were testing on animals, testing lotions and deodorants, and these animals actually developed cancer and died. In some cases, what they did is that they then took out the animals that died, the sick animals, and they replaced them with healthy ones. And what happened with this is that they actually were obviously audited, and they found out that their data was just too good. Yes, we all want to get an R-squared value of 0.99 or 1. But in some cases, like I mentioned in the first slide, science is about testing, because any data you're going to generate has to be reproducible. If you give me a protocol, or I give you a protocol as a senior student, I'm giving you a protocol that you can reproduce. You can give the protocol to 5 of your colleagues, they should be able to reproduce it. It doesn't mean each of us is going to get the same value, because remember, machinery is different, there's a slight difference in buffers, but it has to fall in a particular range. And if we're finding that all 5 or 6 of us are getting brilliant, 99.9% confidence interval, that actually becomes suspicious. So when they were actually audited, they found that aside from replacing the sick animal with healthy ones, they were actually doing a lot of their studies on decomposing dead animals. And that's where it was like, okay, that's actually problematic, because now you're saying that this lotion deodorant is good. And we know this, we see this in industry, where suddenly 6 months down the line, I mean, what was it, a few years ago, we couldn't have, what was this? What did we have? We couldn't have processed meat. Yes, we couldn't have processed meat because it had poisoning with this. I think last year, there were certain cans of food that had to be recalled because there was some problem with them. Apple juice also was problematic, I think about 6 months ago or 3 months ago. Do you see what I mean? It's when you have fraudulent data. This is why. And sometimes it's not that you're sitting out to be fraudulent. You don't keep good notes. You don't plan your work. You get this amazing data, and you yourself don't realize that maybe you added milligrams of a compound instead of micrograms. So you added milligrams of a compound to your cell line, for example, treated your cell line, and suddenly got 99% of your cells dying. And we want that. We want in cancer studies to have a compound that kills most of our cells. But remember, it's in your body. So toxicity, milligrams is way too high. And you will not even discover that you've made a mistake like that until maybe a senior person tries to reproduce it or checks your stuff and realize, but wait a minute, when you did the calculations, you didn't divide by 1,000. So you're actually working in milligrams of compound instead of micrograms. Do you see what I mean? Compound effect. And this is why you have to get very comfortable with good laboratory practice and calculations. So that was what happened with them. And there was a whole case. It was a federal trial, and they served jail time, so on and so forth. So what are the objectives? I've already spoken about all of them. It's to make sure, and I've said this already, is to make sure that every step is valid. So that means, firstly, my test system, my protocol. Is my protocol up to scratch? Has it been optimized? But in addition to that, the equipment that I'm going to use, is there a standard operating procedure? And some of you that are in lab already, you will notice that most of the equipment has SOPs. And the reason for that is if you're using equipment to quantify DNA and I'm using it to quantify DNA, we both need to be at the same level. So that when you present 0.5 micrograms of total DNA and I present 1 microgram of total DNA according to my protocol, we can then compare and see how to improve the protocol. So you see why SOPs become important. And then archiving. Remember, like I said, very few people are doing novel research because research is moving at a very alarming rate because of the technological advances. We need to archive our records and materials for many reasons. The reason for that is I'm archiving it so that the next student that comes along doesn't have to start from scratch. Okay, someone else has optimized the protocol for you, especially if you have a senior student or a postdoc, they've optimized it. Their job is to teach you those skills. And that's even if you decide to stay in academia or you decide to leave. That's where it becomes important. Quality assurance to ensure that we're reporting the studies. So this is why GLP is important. So again, I think we can all read this. We start with the investigator, quality assurance, make sure that the SOPs and these controls, and make sure that the equipment is always calibrated or you know how to use the equipment properly. I mean, you sometimes find something as clicking something wrong on a plate reader and reading a absorbance at 520 when the protocol clearly states to read it at 620 will throw out your absorbance readings, which then throws out your standard curve, which then throws out your total amount of compound. Do you see what I mean? Compound effect. So that's the first thing. And then secondly, I think a lot of students get overwhelmed is that you need to understand that research is now collaborative. Whether you are honors or masters, you're going to be working in a team. If you currently belong to a research group, you're not the only one. It's very rare. You have a few master students, you have a few PhD students, and you have a few postdocs. So the idea is to understand it's collaborative, so teamwork. So as much as we all come with an individualistic approach in how we're writing and so on and so forth, you need to learn to work with other people. And that's the problem that a lot of students struggle with. Something as simple as cleaning up your bench after you've used it, making up the ethanol. You learn that, and that's when you have very good community, and also you're able to have a good collaboration with your colleagues. And the key thing, the reason why we focus on the collaborative work is that, remember, sometimes if you're an honors student, even or a master's, the data that you are generating is kind of like a pilot study, which will then go towards somebody else's. So if you're working on a particular study, it might form part of a bigger picture. If you belong to this particular research group, that particular PI has a particular niche field in which he or she is carrying out their research. Your study falls part of a bigger study, which then falls under an umbrella. And so as a result of that, you need to understand that when you're collecting your data, when you're looking at everything, making up your buffers, it's part of a bigger picture. And that is why you have to understand collaborative and why GLP is important. Okay, so that's on GLP, which I think is common sense, but not necessarily. It's understanding that if I'm required by Friday, for example, to have a particular assay done, where do I start? And a lot of students don't do this. You see them struggling at PhD level as well. I start on a Monday morning or a Sunday, whatever works for you. Monday night, some of us are late, prefer working late. Doesn't matter. Idea is planning. Open up your lab book and plan. I'm going to make up my buffers. I'm going to do all my calculations and make up my buffers on a Monday. On a Tuesday, I'm going to then make sure everything is sterile, and I'm going to seed my cells. So I need calculations done for those. On a Wednesday, I'm going to do the incubation. On a Thursday, I'm going to then collect, read, and on a Friday morning, analyze the data. Rinse, repeat, rinse, repeat. And that helps. Some students arrive and then only read the protocol on a Tuesday, 2 hours before their cells are ready. Do you see what I mean? And then make mistakes, and then you actually run around in circles by yourself. So you'll find like something that could have been fixed in a week is taking you 4 months. And that's how you have students that are getting, not always, but some students are getting into 3rd or 4th year. Because of that, it's understanding that you're going to bump your head constantly if you do not plan and have good laboratory practice. Okay, so I sound like I gave you a lot of like speeches. I'm done with that now, so we can move on to calculations. So again, calculations. Are there any questions on good laboratory practice? Are we awake? No questions? Okay, I'm going to take silence as no questions. Let's get to calculations. So before you get to calculations, I always tell students, let's go back a bit. It's something as simple as understanding the metric system. I'm not going to go through all of it because we all know this already. This is from 1st, 2nd, 3rd, metric, so on and so forth. I just quickly want to touch on a few things that become very important in your calculations. Understanding units of length, mass, volume, and amount, and being able to convert. So what does that mean? One thing that you need to get comfortable with as well is the metric system. So what are the decimal places? What does it mean to go from a millimolar to a micromolar to a nanomolar, or a milliliter to a microliter, and so on, a microgram? You see what I mean? And that's the way you have to understand your metric system quite well. And that's something as simple as that, dividing by 1,000, dividing by 100,000 or a million. That is what tends to inundate students. So a recap of that quickly. We all know kilo, okay? That's 10 to the power of 3, okay? Deci is 10 to the power of 1. Centi, 10 to the power of -1. Milli, 10 to the power of -1, sorry. Milli is, we know, is 10 to the power of 3, so that means it's 0.001, okay? And then we have micro, which is 10 to the power of -6, so it's point, and then there's about 5 zeros before the 1, okay? And then nano. And that's something you have to understand, because sometimes you will have to make up something that is going from grams to micrograms. or goes from grams to milligrams to micrograms. And you'll see that when you have a particular compound, you'll be given a compound that's in 1 gram. You can't treat your cells with 1 gram. Neither can you treat your cells with a milligram. That's too much. Okay, 1,000 milligrams makes up 1 gram. So you have to then go from 1,000 milligrams down to micrograms, because a pharmaceutical level requires you to get 50 micrograms per liter in order to be at pharmaceutical level, so that it's not too toxic for your cells. Do you see what I mean? So you need to understand interchanging between grams, micrograms, nanograms, and so on, and also going from milliliter to microliter, and so on and so forth. So that is where I say you have to understand your units of length, units of mass, units of volume and amount, and being able to convert between them. It makes it much easier for calculations. So one of the things we're interested in concentrations. That's why we all start. You go to a lab, you need to carry out a particular assay. Before you're carrying out an assay, you're going to have to make up the buffers. Sometimes the buffers are made up for you. Sometimes you have to make them up on your own. You have to get comfortable with calculations. And we start with concentrations, because remember, I have a powdered form of anything. I need to then dissolve it and then have it at a particular concentration so that I can work with it. So concentration is the amount of solute in a solution, and we can make it up a certain way. The one that we're most familiar with is weight to volume. Okay, so we're expressing it. The concentration is expressed as the amount of solute at a certain volume. So it's either your milligram per mole, your gram per mole, or your microgram per mole, per milliliter, sorry. So yeah, and that's when you're making up your compounds, you're making up your drugs, you're making up whatever you're making up. Often we work in milligram, microgram, sorry, yeah, per milliliter. So if I have 5 milligrams of a compound, which is my solute, I dissolve it in 10 milliliters of water. I then have a concentration of 5 milligrams per 10 moles, but you want to have it both uniform. So it's 0.5, you divide it by 10, having 0.5 milligrams per milliliter. Okay, and then some of you work with molar solutions. Okay, and a molar solution is where the concentration of the solute of your compound is expressed in number of moles. And for you to have that, you need to have your molecular weight or your molar mass. And this is where students struggle with, because they don't understand what that means. So if I'm making up a buffer, where do I start? Your supervisor is like, okay, you need to do an assay, you need tris buffer. What do? You first go and get powdered tris, and you look at the molar mass, which is on the bottle. It's written there in grams per moles. That's your molar mass. And then you need to make up a solution. So you first have to calculate the number of moles of the solution that you need. And that will be your grams. Okay. So to find the number of moles, that's the grams of the solution divided by the molar mass. Okay, I'm not going to go into depth with that one, because I think most students do that in chemistry quite a bit. I'm more interested in the other ones that we do in lab. So that's the first thing you do. I take a bottle in powdered form, I weigh out a certain amount of grams, divided by the molar mass in order for me to get the concentration, the number of moles that I need. And from the number of moles, I can then dilute it down and then make up my molar solution. And the reason for this is that when you're in lab, you need to have a stock solution, and then from a stock, you dilute from a stock into a working solution. And that's the thing students don't understand. When you start anything in lab, again, systematically, you start with a stock solution, and from a stock solution, you dilute down to a working solution. My working solution is where I will make up my multiple concentrations from. And this is why you have to be comfortable with calculations. So solutions are usually prepared for certain, because we need the particular molarity by calculating the amount of solute needed, okay, to prepare a particular volume. So if I want to make up 1 molar solution, 10 moles of a 1 molar solution of sodium chloride, how much of the powdered form of sodium chloride do I need? So then that's the first thing. Then what do? As I said, you take this sodium chloride, you weigh it out, and then I dissolve it in water or in buffer or in alcohol or whatever, but I dissolve it in the solvent. And then I get a final solution at the desired total volume. So a quick example, put that in context. I am making up a 0.5 liter, okay, of a 0.25 m molar solution of calcium chloride for my assay, which has a molecular weight, grams per moles, of 111 grams. Okay, how do I make this up? Very simply, I'll calculate. The example comes afterwards. It's 0.5, remember it's liters, okay, times 0.5 mole of the solution divided by your liter, so it's the L, because remember, you need to make sure, if you go back to this, the conversion of the units, you need to always make sure that the units are the same. So if you go back to this example, we dissolved 5 into 10 like that, but we present it like this. So we divide, so that's all uniform. So that's the same concept here. So I have 0.5 times my 0.25 divided by the L for the liters, so they cancel out, okay, times 111 grams, which is the molecular weight, divided by the mole, so that the mole is canceled out, and then I will get it in grams. Okay, so if you do that, it's a simple calculation. It will be 0.5. Okay, let's see if I can just, doesn't keep up. Okay, my screen is frozen. Okay, let's go to the calculator. This is something students have been struggling with. Okay, so I'm 0.5 liters times 0.25 moles times the molecular weight, which was 111, okay, gives me 13.875 grams. So if you go back to that, what is the amount that I need? I need 13.88 grams of calcium chloride in powdered form. Most of them are from Sigma with the red cap, to make up 0.5 liters, which is 500 moles of a solution that is at a concentration of 0.25 molar. Okay, but in some cases, maybe some protocols might not give it to you in molar, they might give it to you in millimolars. Okay, so I then need to take this and change it. So if I have a 0.25, and you'll see this when you look at a lot of manuals and protocols, sorry, they'll give you something maybe in M, which you then have to change. And this is why I said in the first slide, you have to get comfortable with conversions of units, because you constantly have to change from 0.1 to 0.001, 0.0000. You know what I mean? So you have to get comfortable moving between the two. So if I now want to present this 0.25 millimolar molar as millimolar per liter, how do I do that? I then take it, so it will be 0.25, okay, divided by the liters, because remember it's in liters, times 1,000 millimolar over 1 molar, okay? And that will then give me 250. So then can you see you're dividing by this 0.25 is just being divided by 1,000 to get it into millimolar, okay? Now, this is not a calculation you actually have to write out, because when you get comfortable with calculations, these are calculations you can do in your head. Why can you do them in your head? Getting comfortable with the metric system. If I know what micro is, it's to the power of this, a -3, -6, -9. I automatically know what I'm dividing by, so I can do this in my head. So I know that 0.25 molar is equivalent to 250 millimolar. Just like I know that 5 grams is 5,000 milligrams. Do you see what I mean? The idea as a researcher when you're entering is to get comfortable with these conversions in your head. Okay, so that's B. The example is written out. I just wanted to guide students through this. And then the second one is, how many milligrams of calcium chloride, okay, would I use to make 5 moles of solution? Okay, so remember the initial one we did was we multiplied 0.5 liters times the concentration times the molecular weight. But now this was in liters. This is 9 milliliters of solution. Gets a bit tricky. You see how conversion of units is important. How do I do this one? Similar concept. I have 5 moles this time. I just write it as 5 times 0.25 m, okay, times the 111. However, because I'm in moles instead of liters, I divide by 1,000. Okay. And in addition to that, I want to present this in your milligrams. So I multiply it by 1,000 milligrams as well. Okay. So let's get to this. Okay. Let's see. So that was the first one. Okay. That was very easy to do. The second one, which again, you can do this in your book until you get comfortable with moving from moles to millimolar and so on. But eventually you will get to a point where you are so comfortable doing this in your head, you know automatically when you look at 0.25, what does it mean? Okay, so you can convert it. So that was the other one. How many milligrams, because I have to have it in milligrams, how many milligrams of calcium chloride would you use to make 5 moles of solution? Now in the first example, it was liter and grams. So I didn't have to divide or multiply by 1,000 to convert it. But in this case, I'm dealing with milligrams and milliliters. So I have 5 moles of my solution. And then again, same concept, 0.25 moles. Multiplied, in this case, we're dividing by 1,000 milliliters because we're converting it. Okay? Multiplied by your molecular weight, which is 111. And in this case, I do not want to present it. Here, I presented it in grams. I want to present this in milligrams. So I'm multiplying by 1,000. Because why? 1 gram is 1,000 milligrams. So if I want to convert grams to milligrams, I need to then multiply or divide, depending on how I'm going. So I'm dividing. Okay? And then I multiply, in this case, to get into milligrams. But because I have milligrams and milliliters, I also have to divide by 1,000. Whereas here, it's liter to gram, so I didn't divide by 1,000. Does that make sense? And that again goes to the concept of you have to make sure that there's uniformity. If it's milligrams, it must be milligrams per milliliter. Okay, if it's nano to nano. In some cases, it will be slightly different, but you have to take your conversions into account and either divide or multiply. So in this case, I'm dividing by 1,000 to get into milliliters because it's 5 moles. And I'm multiplying by 1,000 to get it into milligrams instead of grams. And then you have 138. Okay, so how do we do it? I'll go through a few of these. How many grams, this is better to do as a tutorial, but let's go through them. How many grams of sodium chloride with a molecular weight of 58.44 should you use to make up 100 moles of a 0.37 solution? Okay, so if you go back to the previous example, if it was in liters, you would do it this way. So it's 100 moles times 0.37, okay, times 1/1,000. You understand why it's 1/1,000? And the reason why it's 1/1,000 is because it's 100 moles. Okay, times again my molecular weight. So again, similar concept to this one. Okay, but because it's not in liters, it's in milliliters, I would then have to multiply, well, divide by 1,000. Sorry, let me not say 1 in 1,000, might be confusing. I'm dividing everything by 1,000. So 100 moles times 0.37 concentration times the molecular weight, but I'm dividing everything by 1,000 because I want to change it from liters into milliliters. Does everyone understand that? So what do you come up with? Has anyone got a calculator? Does anyone want to take a shot at this? Let's do it in the calculator and see. 100 moles times 0.37 times the molecular weight, which is 58.44, divided by 1,000 to get into milliliters. There we go, by 1,000. Okay, so if you come back, how many grams? I would then divide it by 1,000 to get it into grams, so it would be 2.1 grams. Okay, again, similar concept would this be. How many grams of sodium chloride should you use to make up 37 of a 1 molar solution? 37 moles times 1 m times 58.44 divided by 1,000 to get the same 2.1. will also give you the 2.16 grams. So then if I'm in lab, I know that for me to make up a 1 molar solution, I only need 37 for my study, or I only need 100 for my study, depending on what I'm doing. I need 100 moles of a 0.37 molar solution. How many grams do I weigh out on the fine balance? 2.1 grams. Okay, so same concept. Whereas here, for example, how many grams of sodium chloride should you use to make up a 5.8 liters of 3.7 millimolar? So that's why I said, remember, you have to get comfortable with conversions. 3.7 millimolar is equivalent to 0.0037 molar. So then I'm able to do a 5.8 times 0.0037 times again. So go back to the example. So I'll do 5.8 liters times 0.0037 molar, because I converted it already from millimolar into molar, okay, times your molecular weight, which is 58.44, which will then give me 1.25 grams. Okay, that's how you will then get that done. And then all you're doing with this, and this is the thing, you only need to learn 3 equations and learn how to then solve for X. And I think we have done this in introductions. All you're doing is you're learning 3 equations and solving for X. If you go back to this, you're either going to solve for volume, solve for concentration, and solve for this. The molecular weight will always be at the back of the bottle. For any reagent, there will be a molecular weight. Okay, so obviously with your nanoparticles slightly different, but generally with any compounds or drugs or chemicals that you're using, there will be a molecular weight. So this will always be the guide you. So then I need to calculate either one of these. Number of moles will be the grams divided by the molar mass. That will give me the number of moles. I have that. Liters, how much do I need in lab for my stock solution? I want to make up a stock solution of 5 moles. There, 5 moles is my stock. So I'm solving for X. You see what I mean? So the idea is, you don't have to go through all of them. The idea is always to solve for something. So in this case, we're solving for mass, but we have to make sure that the conversions of the unit are correct. So in some cases, it's worth liters, okay? And in some cases, it's worth milliliters. If it's milliliters, I'm dividing by 1,000, okay? And if it's a liter, I'm not dividing by 1,000. But if it's millimolar, I would then have to change it into molar, which would then make it 0.037 millimolar. And then I'm getting the mass. And in this case, it's slightly different. You have weighed out 5.84 grams, okay, of sodium chloride. What is the total volume to make up a 1 molar solution? So we're going here now, again, similar concept. I now have this. I know this. I know this. I'm solving for this. Do you see what I mean? And this is why I say you need to get comfortable with decimal places, 0.1, 0.00, and how to either divide by 1,000, multiply by 1,000. That is the one thing to learn. And the other thing to learn is 3 equations and solve for X, whatever you're trying to answer. That will guide you through any calculation that you need to do. Okay, so if I've weighed out 5.84 grams and I have a total volume of that, how do I do that? I'm solving for X. So if I go and I solve for X over here, okay, it's now 5. It's very hard to do this over. I would prefer a dark cam, but anyway, 5.84 grams. The molecular weight is 58.44 over the moles, okay, times 1,000, because it's 1 molar. So then solving for X, you then change things around. Okay, so this will then go to that side and so on. I think you guys know how to do that already. That's math where you take things over and then it changes. So then to solve for X, I'm going to then have 5.84 grams, because remember it was on the equal side, okay, divided, because it comes over, so it changes from a multiplication to a division. So divide by the molecular weight, so it's 58.44, okay, and because I wanted in milliliters, I then multiply by 1,000. So I will then have 99.9. Okay, let's go back to the example. I have 5.84 grams of sodium chloride, which has a molecular weight of 58.44. However, I don't know how much of this I need, but I know that I need a concentration of 1. But I don't know how much I need. So then I take 5.84. If I go here, and I'm going through this slowly because this is where students get slightly confused. 5.84 is here. The molecular weight of 58.44 is here. The 1 molar is here, and that's the X. Okay, so what do? I take it over. So if I take this over, it's multiplying here, it needs to then divide. So that's why it will be 5.84 grams divided by the molecular weight of 58.44. Okay, and then because this is also dividing, it's times 1, that's I left it out. It is times 1, so it's negligible. I want this in milliliters, so I multiply by 1,000. And that's how then I will need 99.9 moles of my solvent, water, media, whatever, with 584 grams of sodium chloride to give me a solution with the concentration of 1 molar. Okay, and then the other thing, let's quickly, before we move to dilution factors and the most important thing, standard curves, because I want to do an example, is sometimes you need to do percentage volume. So this is weight to volume. Now we're moving to percentages, so that's out of 100. So if I have a 250 moles, okay, volume, final volume is 250 moles, and it needs to be a 5% solution. This is, you see this buffers are 10%, buffers are 5%. How much sodium chloride would I need? So we know 5% is 0.05 already, okay? I multiplied by 250 moles. That's how you make up a solution. If you're told to make up a 10% solution, and you'll see that sometimes the senior student will be like, go make up a 20% solution. Okay? If I'm making up a 20% solution, I need to decide, am I making up a liter of it? Am I making up 250 because I want to have my buffers fresh every week? But you need to decide what the volume is, and then you just do the percentage. So in this case, it's 250 moles of a solution and a 5% solution. So that's 5%. So that's obviously 0.05 times 250 moles, because that's my final volume, will give me 12.5 grams. Do you see? So therefore, I need 12.5 grams of my sodium chloride dissolved in 250 moles of my solvent, water, which will then give me a 5% solution. And why this kind of thing stumps a lot of students? This one stumps a lot of students because sometimes when you're making up a particular buffer for a experiment, the buffer has many solvents in it or many solutions. So maybe if you're making TBE buffer, if you're running gels, it's made up with tris, it's made up with what is it made up with? It's tris and something else. It's a whole lot of things. I haven't been in lab in a minute. It's made up with different types of things. So it might need a 10%. Okay, if you're doing Western blots, so you're doing protein work, you might need a transfer buffer. Transfer buffers are usually 20% methanol, 10% tris, and then another percentage of something else. So you have to make up each of the solutions separately and then put them together to make up your transfer buffer. Do you see? And this is what some students, when a senior student will be like, go make up a 20% solution. How, why? First, foremost, how much do I want? A liter is the easiest, 1,000 is the easiest. So you make up a liter in a shot bottle, 5%. 5% will give you that. So in this case, we weighed up 250. 5% of 250 gave us 12.5 grams. So I weigh out 12.5 grams of sodium chloride to give me a 5% solution. Okay, let's continue. Cell dilutions. Now, remember, as I said, you're going to have your stock solution and you're going to have your working solution. So as a result of that, you have a stock which is very concentrated, and from there, you're going to dilute it down accordingly for a particular experiment. And that's a standard thing. You don't start with working solutions. There's no need for you to have 10 tubes and do 10 calculations. You do one calculation, make up one stock. And from the stock, you're diluting down. So as a result of that, you need to, and this is because you're going to determine the amount of material in an unknown solution, and you're going to test at different dilutions. And this is again in the lab as well. You're going to have a stock of a particular compound, a particular drug, and you're going to dilute it to find a concentration range. So as a result of that, you have to dilute it. So how does dilution factors work? It's my initial volume divided by my final volume. That's my dilution factors. My initial volume divided by my final. So if I have a 2.5 moles, okay, and I dissolved it in a diluent, in 7.5 moles of diluent, water, for example, I took 2.5 moles of my stock. I dissolved 2.5 moles of my stock solution in a diluent, i.e. water or media, to make a final volume of 10 moles. What is the dilution factor of that? And again, this is the key way to work it out. The initial volume, 2.5, divided by the final volume. What is the final volume? 10. Because why? 2.5 moles of stock plus diluent gives me final volume. So this is where students also get confused. The final volume is taking into account the amount of diluent that you're adding, the amount of water, the amount of media to make up the volume. So in this case, it's 2.5 in a final of 10 moles. And that will give me a dilution factor of 1:4. That's the ratio. So it's a quarter. Does that make sense? So this is where dilution factors get tricky for students. So, and then further, what gets even more tricky is that if I did 2.5 into 7.5 to give me a final volume of 10, that's a 1:4 dilution. But we do often in lab sealed dilutions. So there's types of dilutions. We do a log dilution, and this is if you're doing bacterial work, especially, you will take 1 mole, okay, of 1,000 colony forming units, dilute it into 9 moles of your diluent to make up a final volume of 10. So that means you're taking this 1,000 colony forming units, okay, and you're dissolving it by adding 9 moles to it, which means the concentration changes. So it's exactly the concept of OROS. You're starting with a concentrated OROS and you're dissolving it down, which means it's not going to be as sweet, isn't it? Similar concept. As I always tell students that, you know, sometimes just simple examples help. I'm taking my initial sample, which is my stock, and I'm making a 10-fold, which is a log. So it's a 1:10 ratio. Okay, why is it a 1:10 ratio? 9 plus 1, 10 moles. So 1:10, because 1 represents the amount of sample added. So from my stock, I took 1 mole, I added 9 moles of media, broth, water, whatever my diluent is according to my study, and then I made up a final volume of 10 moles, which means the concentration was diluted from 1,000 colony forming units per mole to 100. And this becomes important when you look at the protocols for carrying out particular experiments. You need to have a final working sample. And this is how you need to then understand the concentration. So this is your log dilution. Okay, so this is usually commonly seen in your bacterial work, because if you have 1,000 colony forming units, and you spread plate this from your stock, which means if you need to carry the experiment for 24, 48, or 72, depending on your study, you're going to flood the plate. Do you see what I mean? So that's why you have to dilute it down so that you have enough colonies, but also at the end of your experiment, you don't have an saturation. And I'll explain how that becomes a problem for standard curves. And then the common thing that we're all familiar with is standard sealed dilutions, which get very confusing when you have a 96-well plate. Okay, so like I said, similar concept. I start with my stock 10 times or whatever, and I'm diluting down. So if I have 1 mole of a solution, okay, I have 1 mole of solution, I'm adding 2 moles of water. Right, let's start there. Let's go slowly. Let me just do this, it's much easier. Okay, if I have 1 mole of a solution and I'm adding 2 moles of diluent, before we continue, this makes a 1:3, right? Because initial volume is 1 mole and my final volume is the 2 plus 1, which is 3. Do you see that? And I think this is where students get confusing. And the idea is to then, even if you're still diluting, go back to this little calculation. Initial versus final gives me the dilution factor. And if you do that every time you dilute, eventually you will get comfortable with sealed dilutions in your head. So 1 mole plus 2 moles gives me a dilution factor of 1:3 based on this calculation of dilution factors, right? From this, over here. Okay, let's do it. From this, I take out another mole. Okay, there's an example coming. I take out 1 mole of this, and this is where students get confused. I'm not taking out 1 mole from here. No, I'm taking out 1 mole from here. I'm serially diluting. So if I look at a plate, I'm going from A1 down to B1, down to C1. So I keep on taking out of each. So I'm diluting Oros. So again, I take a cup of Oros from the bottle. I put it in 1 glass. I take from there half a glass and put it in another glass of water. It's too sweet. I then take out from there into another glass and add more water until I get to a place where it's comfortable for me to taste. Similar concept. And this is where a lot of students, it's something so simple, but really inundate students. This third 1 mole here, I'm taking from here. So we continue. 1 mole is taken from this initial, is then added to another 2 moles. So I take 1 mole of a third dilution factor. I add 2 moles. Okay, that gives me a dilution factor of 1:9. Does that make sense? Because I'm then multiplying the dilution factors. So in this case, I have 1/3, and I'm taking 1/3 of 1/3. Does that make sense? Because I'm taking from this one. I'm not taking from this one. I'm taking here. So I'm taking 1/3 from the 1/3, which gives me 3 times 3 gives me a dilution factor of 9. And then I continue. I'm taking 1 mole from here, adding another 2 moles to make a 1/3 dilution. So therefore it will be 9 times 3, which will give me a final 1:27, 1/27. Do you see? So the key thing that students don't understand is that you're serially diluting. I'm taking from the initial stock, and I'm going here, and I'm taking out from there, and I'm taking out from there. And that's the key thing. So let's do a quick example. Suppose I have 1 mole, okay, of a sample, and I'm adding 9 moles, okay? I then make 4 seal half dilutions, 1:2 dilutions. What is the final dilution factor? Now, the key thing to understand is that you must always multiply because you're trying to, it's doing sequential dilution. So the idea is if I have 1 mole of sample plus 9 moles of water, from that initial calculation I showed you guys, what is it? The dilution factor is 10, okay? It's 1/10 because it's 9 plus 1. I'm making 4 half dilutions. That means I'm doing a, okay, 1/10 was my initial dilution because of 1 plus 9. I then have to do 4 seal dilutions. So that's 1/2, 4 times. So I'll write this out better when I actually make a PDF for you guys. What will that give me? So again, a similar concept. I had a 1/10, right? I'm taking half from there. Then I'm taking half from that half. And then I'm taking half from that half, and so on and so forth. And this is how you have an 8-fold, 4-fold, 10-fold. So my final dilution factor will be what? Similar concept. The same way I multiplied, I'm doing the same thing. So I will take 10 multiplied by 2, and then another 2, and another 2, and another 2, until I come with a 1:160. So if I have a 1/10 dilution of a sample, and I make 4 1:2 dilutions, seal dilutions, the final dilution factor will be 1:160. Similar to the concept here. We took a third of a third of a third. We came up to a final dilution factor of 127. I know it's a bit confusing, but back to another example. I have a mole, tube 1, 0, because it's 1 milligram per mole of concentration. And then I'm diluting by a half, then a quarter, then an eighth, then a 16th. So again, like the concept of Oros, I'm starting with a concentrated Oros at 1 milligram per mole. I then take half of this in a tube, in a glass. I take 1 mole, which means I'm halving the concentration. Do you see what I mean? The volume stays the same because even though I took 1 mole here, okay, I'm going to add 1 mole of water to make it up for 2 moles throughout. It has to be 2 moles throughout. So even though the volume will always be 2 moles throughout, the concentration will change because of the dilution ratio. Does that make sense? So in this case, an easy example, I started with 1 milligrams per mole. I took 1 mole from this solution. I added another 1 mole of water, which means I diluted it, which means the concentration now is 0.5 milligrams per mole, which is, if you go to conversion, 500 micrograms. You see, so 0.5 milligrams per mole. I then took from the 0.5 milligrams per mole. I took 1 mole. I added another 1 mole of water. So I'm going down from 0.5 milligrams per mole. I'm going down to 0.25 milligrams. That means it's a quarter. So 0.25 is a quarter of 1. Do you see? Which is again equivalent to 250 micrograms. I take 1 mole of that. I go further down. I'm 0.125. This is what CO dilution is. It's a 2-fold CO dilution. Some cases will be 8-fold. That doesn't matter whether it's a 2-fold, 10-fold, whatever fold. The idea is to always know your ratio of each of the tube, each of the well, and that ratio allows you to figure out how the concentration of that initial stock is being diluted. And this is how you then, again, similar concept of, I have a drug that I want to test. I made up a stock solution of 1 milligram per mole. 1 milligram per mole means that it's 1,000 micrograms. Do you see? My supervisor says that he read a paper, and the paper says that, you know, if I dilute from 1,000, which is 1 milligram, 1,000 micrograms, I go down by 100 until I get to 0. I can get a very good concentration range to see how the drug will work. So that means I need to dilute it from the stock. I dilute it by adding more media to different working solutions, and I will go down from 1,000 micrograms, which is 1 milligram, down to 900, 800, 700, 600 in a particular volume for the amount of cells. So you see, this is all why serial dilutions become important. Okay, now moving on to standard curves. Why do we need standard curves? Now, sometimes you do this in the lab, but in some cases you don't know the particular concentration because you're trying to do either MTTs, so absorbance becomes important. So we use standard curves to show the relationship between a particular compound and the absorbance, because each time when you're treating something, it will have a particular absorbance reading. You can use the absorbance reading to calculate the amount of that compound at that particular concentration range. So this is where standard curves become important. In some cases, you will do it using a compound or a known compound, which acts as your reference, okay, or a blank. So a reference solution is something that lacks the compound of interest, or in some cases use a blank. In lab, it's very rare that you use a blank, you use a reference. And the most common one we use, especially for protein work, is your BSA. So what you do is you plot a curve. So how does that work? I'm going to do an example. So before we get to plotting a curve, is that you're going to have something like this. The amount that you added to your cells. So like I said, your supervisor told you, here's a stock of a particular compound I ordered from wherever, of 1,000 micrograms. I want you to go down by 10 or by 100. So if you go down by 100, it's 900, 800, 700, so on and so forth. So you get to 0 to see what effect it will have on a particular cell line for you to get a concentration, dose response. So first, you then run an MTT, for example, whatever assay you're doing. Your 900 is the amount of reactant, amount of compound, and each one at a particular range will exhibit absorbance. You plot that together and you will get something like this, which is known as a standard curve. So you sometimes get a completely straight line. In some cases, you don't get a straight line. And the reason for that is you will have saturation. So for those of you who are in lab already, you will find that at a particular concentration, you will get the same absorbance reading of 0.9, 1.2, 1.2, 1.2. It doesn't matter. What the concentration is here on this particular axis, it will always be 1.2 absorbance. What that tells you is that your compound with your reagents has reached a point of saturation. So it's a saturation of absorbance. So that's why you don't read on the non-linear, because now it's a non-linear range. It's an invalid range. You're interested in the linear portion. The linear portion means with the increase in compound or drug or nanoparticle, there's an increase in absorbance. So that gives me a standard curve that I can work with. Okay, and the reason for this is that if you work in a linear range, this is a bit of maths, you're coming to the concept of y equals mx plus c. Remember, linear graphs are given as a y equals mx plus c, where c is your y-intercept, which is here. So if it's a 0, 0, it will be, your y-intercept will be 0. So that is it. Once you've created a standard curve, okay, of your unknown solution, you look at the linear range, and I'm going to do an example of this. You can read all this up in your own time. Okay, that will allow me to have a linear graph of y equals mx plus b plus c, whereas c, b, whatever you want to call it, the y-intercept is 0 because the line goes through the origin. It's linear because the increase in my drug or my compound results in an increase in absorbance. Okay, so it's a corresponding amount. Okay, and so therefore you have this equation. Now, some people in lab, when they have a linear curve like this and they want to find a concentration, they just plot it like that on the graph. We don't do that in lab. In lab, we use this equation. So this is the mathematical equation that we use. Y equals mx plus c. And by doing that, okay, we use the standard curve to create a relationship between the absorbance and the amount, because it's in a linear form. So let's go to the example. So I need to make a standard curve of a compound by setting up a series of tubes containing various amounts of compound Z in micrograms. And I need to measure the absorbance of each tube. Okay, so you see, I have to put everything together, all my abilities to calculate together. And this is why my seal dilutions come into play. Okay, so if I have an example of Z in micrograms, I have to measure the absorbance of each tube. Suppose you then determine the slope of the line, and I get an equation of y equals 0.0262 and times x, x being the concentrations. We don't have the y-intercept because remember it's a linear graph. So y is negligible. The y-intercept is negligible because it goes to the origin, it's 0. I'm going to do an example in Excel to make it much easier. If you then measure the absorbance of an unknown solution containing Z, I find that the absorbance is 0.198. I then need to use that to calculate the number of micrograms of Z in this tube. So tube 1, okay, gave me an absorbance reading of 0.098. Okay, I then have to use that to calculate the number of what is the actual concentration using that. So if I have y equals 0.062 or 0.3, okay, times x, again, it's conversion. Remember, as we previously did, we're solving for x because I already have my absorbance. So I'm solving for x. If I'm solving for x, that means if y equals 0.98, this is y, okay, this 0.62 is multiplying here, then needs to divide. And because it needs to be micrograms, I'm looking at it in a total of 1 microgram. So it's 0.98 divided by 0.62, solving for x. So in tube 1, it's 3.19 micrograms of compound that's been added. So let's put this in context. I did a experiment in the lab, okay? I started with a stock solution of 1 milligram, okay, 1 microgram. Let's make this milligram. Let's make this milligram, sorry. Okay, I started with 1 milligram. That was my stock solution. So if I go back to the presentation, I started here. I was given a stock solution of 1 milligram per mole, and I now need to carry out that. I can't add 1 milligram to my compound, I mean to my cells, or to my treatment as my treatment. It's toxic to the body, it'll be toxic to the cells, so I have to dilute it down. So then I made a dilution by 100. So then I start with 0 because that's my control, cells only untreated. I then did 0.1 milligrams, 0.2, 3, 4, 5, so on and so forth. I treated my cells with this in triplicate, and I found the average. And these are the absorbance I got. With no compound, it was 0.52, and then you can see there's an increase in absorbance with an increase in concentration. This is what the plate reader gave me. Okay, this is my row 1. Okay, A down. Okay, so this is what I get. From this standard curve, I need to find out how much of my compound is in the wells. So first, what do? I need to then plot it. So you highlight this, you insert. Okay, recommended chart, it's always a scatter plot. Okay, here's my scatter plot. I'm not going to correct all the editing of the scatter plot. I'm interested in the values. So I have the values over here. Okay, so we always do it mathematically instead of thingy. So I click on each one. I think we're all familiar with this, and I add a 10 line. By adding a 10 line, which is a line of good fit, I then can have the R equation and the R squared value. Okay. Because it's not at 0, I now have, because in lab it will very rarely start. Remember, absorbances won't start at 0. And for us, the reason why they don't start at 0, unless you take out the blank, is because we have cells. So cells will also admit their own absorbance. So in lab, it's very rare that you're going to have a 0, 0 origin. So your y-intercept will very rarely be 0. But you're using this concept to calculate. So then this is where my absorbance at 0, no treatment started at a concentration of 0.52. So I have that. From here, again, if you go back, it's the same concept. These absorbance here are my y values because they fall on this axis. I have my slope, which is 0.107, and I have my intercept. So what am I doing? I'm solving for x. So again, you play around. I'm not going to give an example because it's a bit too much information. Again, similar concept. I then solve for x, which means this comes over to subtract. Okay. So you plug it into Excel, it comes over to subtract. And then I divide by 0.17 and I'm solving for x. So if you want to quickly do that, you then do equals. Let's do that quickly. My y, which is my absorbance, minus my y-intercept, which is 0.0443 over that, close brackets, divided by my slope, which is 0.10107, and that will give me the amount of drug in that particular well. Can you see? So automatically I've done the calculation. And there we go. And then you can obviously present it properly by your decimal places. There we go. So now I know, if you go back to the example that we did, similar concept, the absorbance reading of the unknown solution was that, and I needed to calculate the amount of the compound in each of the tubes. So that's what I've done. This is what I treated the cells with. This is the absorbance I got. I then used the y equals mx plus c concept, which is mathematical concept because my r squared is close to 1, which means it's quite accurate, to then calculate the amount of the drug in that particular. So you're solving for the unknown. And this standard curve is something you're going to use for everything almost. When you're trying to solve for the unknown. For proteins, you use BSA for that. And this is why students need to get comfortable with standard curves. Whew, here we go. I hope that helped. Okay. So that's the basics of most of your calculations. The key take home here is get comfortable with the metric system. What is 10 to the power of 3? What is 10 to the power of 6? What is 10 to the power of 9? If 1 milligram per mole, what does that mean? 1 milligram is the same as 1,000 micrograms. 1 gram is 1,000 milligrams. 1 milliliter, okay, is 1,000 microliters. Do you see how you get comfortable? And that's where you get comfortable with the metrics and the units of length. You then can learn to convert easily in your head. And then from there, you get comfortable with calculations. The ones that we did. Okay, how do I calculate for grams? How do I find out the volumes? Practice these. Okay, that's the thing with any maths, they'll tell you to practice. Okay, and then after I've made all those little calculations, I must dilute it because I always start with the stock and then I come to working solutions. So that's where I get comfortable with dilution factors. Stock, and I'm going to be diluting down to my 10 tubes or my 10 vials at a particular concentration in triplicate or quadruplicate or so on. You see how it all step by step comes together and gets more and more complicated. It's the same like mathematics. I start with subtraction and division and I end up at calculus. Same concept. I start with basic understanding units of measurement and then I come with dilution factors. So then from this, I then run my experiment and I plot a standard curve in a linear form. Okay. And from that, I use the mathematical equation y equals mx plus c to solve for my unknown. Okay. Because absorbance. And I solve for my unknown, and this is how I get a linear curve. Okay, so it's a lot of things on calculations, but if you learn the step-by-step process of calculations, you're then able to understand how to actually create your buffers, carry out the experiment, solve for the unknown, and plot it. And then from there, it becomes even more complicated because then you move to data analysis, quantitative, which is an up-and-coming workshop. Okay, thank you so much for your guys' time. It was a lot. I think some of you know this already, so it's just a recap of what you've learned. We have a lot of information on various things like quantitative data analysis, SPSS, and so on and so forth. So please feel free to visit the Encanva site and find us on social media. For those of you who are left, and for those of you who watch the recording, please can you follow out, take a minute or two to actually do the workshop evaluation. So I have sent it in the chat box, so you can copy and paste that in the chat box and quickly fill out the evaluation. Okay, thank you so much for your time. If you have any questions, feel free to email me. If you need a consult on calculations or data analysis, literature review, proposal writing, so on and so forth. So yeah, thank you so much for your time. Are there any questions from the 3 people that are left in the presentation? Okay, I'm going to take silence means that it's either a lot and you guys need to do it in your own time, or we understood everything really well. It's a lot when you're not in the lab, but I think in your lab and as you start working with various experiments, you become comfortable with these concepts. And as you do more and more calculations, it becomes easier. So yeah, if there's no questions, I think I'll end the session. And again, feel free to actually send me drop me an email if you have any questions with regards to any of your research. Thanks so much. Okay, you're welcome.